In this lecture, we will examine the pathway that connects glycolysis to the citric acid cycle. Product of glycolysis is pyruvate, and compound that enters the citric acid cycle is acetyl coenzyme A. So conversion of pyruvate into acetyl coenzyme A is biochemical pathway that connects glycolysis to the citric acid cycle. Enzyme that converts pyruvate into acetyl coenzyme A is pyruvate dehydrogenase, and reaction is decarboxylation of pyruvate. So pyruvate reacts with coenzyme A and NAD with the enzyme pyruvate dehydrogenase, and products are acetyl coenzyme A, molecule of carbon dioxide, NADH, and a proton. And that's shown also in this equation. The molecule of pyruvate is converted into acetyl coenzyme A. Coenzyme A is the reactant, product is carbon dioxide, and oxidizing agent is molecule of NAD, which is reduced to NADH. Enzyme is pyruvate dehydrogenase. The conversion of pyruvate into acetyl coenzyme A consists of 3 coupled steps and is followed by the 4th step, and that's regeneration of lipoamide. So the 3 coupled steps are decarboxylation, oxidation, and transfer of the acetyl to coenzyme A. And final 4th step is regeneration of lipoamide. The entire process is catalyzed by the pyruvate dehydrogenase complex, which is composed of 3 enzymes and 5 cofactors. Of those 5 cofactors, 3 are catalytic cofactors and 2 are stoichiometric cofactors. So here is a list of the enzymes. Pyruvate dehydrogenase E1, TPP is the catalytic cofactor. Dihydrolipoyl transacetylase E2, lipoamide is the catalytic cofactor, coenzyme A is the stoichiometric cofactor. And finally, dihydrolipoyl dehydrogenase E3, FAD is the catalytic cofactor, and NAD is the stoichiometric cofactor. The first step of the sequence is decarboxylation of pyruvate. It is catalyzed by pyruvate dehydrogenase component E1. Thiamine pyrophosphate or vitamin B1 is the catalytic cofactor, and in this reaction it is in form of its anion. Anion reacts with pyruvate to give the intermediate, which is decarboxylated, and product is hydroxyethyl TPP. Next step of the sequence is oxidation of hydroxyethyl to acetyl. In this process, lipoic acid plays a crucial role. Lipoic acid forms an amide, and this would be an amide of lipoic acid, lipoamide. In this case, amide is to the lysine residue of dihydrolipoyl transacetylase E2. Now, while lipoamide is attached to lysine residue of dihydrolipoyl transacetylase E2, catalysis of this oxidation step is still catalyzed by the same enzyme as the previous step, pyruvate dehydrogenase component E1. So hydroxyethyl TPP anion reacts with lipoamide, and that regenerates TPP anion, and product is acetyl lipoamide. Reaction mechanism involves nucleophilic attack of hydroxyethyl TPP anion to lipoamide disulfide bond. That results in breaking of disulfide bond and formation of carbon-sulfur bond between hydroxyethyl TPP anion and sulfur of lipoamide. In the final step, bond between TPP and hydroxyethyl is broken, so the products are acetyl lipoamide and TPP anion is regenerated and is able to participate in another catalytic cycle, while acetyl lipoamide goes on further to be converted to acetyl coenzyme A. In the next step, acyl lipoamide translocates to the next enzyme, the dihydrolipoyl transacetylase E2. So this long arm of acetyl lipoamide swings and translocates to the other active site, to dihydrolipoyl transacetylase E2, and there acyl group is transferred from acetyl lipoamide enzyme complex to coenzyme A. This is favorable process, thermodynamically favorable process, because one thioester group is converted into another thioester group. And products are acetyl coenzyme A and reduced form of dihydrolipoamide. In the final step, a lipoamide is regenerated, which means that disulfide bond of lipoamide is regenerated. Dihydrolipoamide arm swings into the third active site, and that's the dihydrolipoyl dehydrogenase E3. There, FAD acts as a cofactor, and in reaction that is catalyzed by this enzyme, dihydrolipoyl enzyme dehydrogenase E3, FAD oxidizes dihydrolipoyl amide to lipoyl amide and therefore regenerates it. Dihydro-FAD is then, in the final step, oxidized by NAD. So it is converted back to FAD, and NAD is converted to NADH. That completes the sequence, and it completes both conversion of pyruvate into acetyl coenzyme A and regenerates the enzyme complex so it can participate in the next catalytic cycle.